I am Anjali Venkateshwaran, and it is my pleasure to introduce Tia Coleman, who will be joining me on today's podcast. Tia is the Assistant Director, Clinical Pathology at Biomere's site in Worcester, Massachusetts. Tia received her bachelor's degree at the University of Massachusetts Amherst and has over a decade of experience as a medical laboratory scientist. So, Joine Biomere is a preclinical CRO with locations in Worcester, Richmond, California, and has multiple sites in China. The portfolio includes comprehensive discovery, ADME, non GLP, and GLP toxicology services. If you're interested in learning more, you can check out Biomere's website at www.biomere.com. So, with that, let's get into today's topic on clinical pathology testing and the development of panels to support in vivo studies. Tia, thank you so much for joining me today. I'm very excited for our discussion.
Likewise, thanks for having me.
Yep. So let's get right into this. So analysis of blood and biofluid samples to identify changes and specific markers, analytes, proteins, what have you. They're very commonly used in the clinic as well as in preclinical studies. So let's start with what's going on at Biomere and what you guys are doing to support in vivo studies.
Sure. So here at Biomere, we have a clinical pathology department with both hematology and clinical chemistry analyzers. We have two clinical hematology analyzers, and the newest of those is the Sysmex XNB 1000 analyzer, which has a very high throughput and very specific platelet markers for more precision analysis. And then we have a Beckman-Coulter DXC 700 AU clinical chemistry analyzer, which also has a very high throughput and allows us to perform analysis on very small serum or plasma quantities.
Wow. No, that's so let's dig a little bit deeper. So you mentioned, hematology as well as, clinical chemistry. So maybe we'll start off with the clinical chemistry bit of it. So, Beckman's got a lot of different analyzers, and you mentioned the AU is, I think, the DXC is the newer system, and the AU is, a little bit older but more suitable for smaller labs. So tell us more about, why Biomere invests in the DXC and kind of how many samples you guys are analyzing or your lab is analyzing on a weekly, basis or daily basis as the case may be.
Sure. So in the past, we had a different analyzer, something called an alpha Wasserman VET XL, and that analyzer required such a large volume of serum that it was really not feasible for us to do very much, especially for any animal model besides a non-human primate. So unless we could provide several hundred microliters of sample for that analyzer, we were not able to get very many analytes analyzed. So we invested in the Beckman-Coulter DXC 700 AU, and we shopped around for a few different options, but that analyzer was able to give us the most precise small volume sample. So, for example, one of the first studies we ran on that analyzer, when we got it up and running, we only needed 20 microliters of serum, and we were able to get two results off of that, which is exactly what the client wanted. And we were able to use a mouse model on that study and also provide interim testing as opposed to just one big bulk testing at the end of study upon necropsy.
Yeah. And that's interesting. So that I think segues perfectly into the something I wanted to talk a little bit more about is typical study design, because I think a lot of the analysis essentially supports in vivo studies. So can you tell us a little bit more about the typical study design that generates the samples for your lab to analyze?
Sure. And to answer, I think I missed one point in your last question too. The other challenge with that previous analyzer is it was super slow. So this
Okay.
analyzer, again, to address the throughput, can sample hundreds of specimens in a matter of hours, as opposed to one or two every 30 minutes on the previous analyzer. So we would load up that old analyzer and walk away for the day sometimes.
Right, right, right.
Whereas this analyzer, I think I ran 80 specimens on Tuesday in about an hour and a half. So very
And
high
wow, and
throughput.
that's fast. And
Yep,
you're, yeah, that is really, and that becomes a big deal because what you don't want
huge.
is, if you have a
The
lot of in vivo
backlog.
studies going, you don't want to back
Yep.
exactly, because
Mhm.
then
That's
yeah, that just
exactly what
becomes
was
a
going
problem.
on. Yep.
Yeah,
And
yeah,
then
yeah, yeah.
the one other thing that I do want to add for that analyzer is an open channel analyzer. So we have the ability to bring on and validate analytes that may not be provided by Beckman Coulter, which is something that we've done recently at a
Okay.
request of a client who was looking for something very specific. We were able to do the
Yeah.
research. We actually found the same exact reagent that was used in a previous study that the client was addressing the same test article or a similar goal for their test article. So we found that same exact reagent. We were able to validate it, bring it on board for them, and give them the test results that they were looking for.
That's amazing. I mean, that to me just lends that kind of customization because I'm sure, I'm sure you've got a lot of regular studies that, that can be addressed with the available analysts, but there's always going to be studies with, custom, oh, it'd be really great if you can add X, or I don't want A and
Right.
B, I want to add this. And so
Yep.
that's amazing. So how does this, customization process take? Is it very difficult to do that on the Beckman Coulter analyzer?
It's not difficult. It takes planning, of course. And to be honest, the more we do it, the more familiar we get with the process.
Yeah.
So
Yeah.
I've done it 3 times now myself, and I've observed them doing it for us in the past. They used to come in and do all of the validation. But when it's a special analyte, such as a non-Beckman Coulter thing, then that's something that we would be responsible for validating. And we've gotten pretty good and quick
I
at
love
it.
it.
As long as we can plan ahead, we're able to perform the work. Um, Again, validate it, make sure that it is feasible for the study in the animal model, and then go from there for study testing.
I think that's a huge benefit because, it's good to have these markers, and some are more informative than others. So if a client program or a specific drug, whatever it is, is attached to a very unique biomarker, then it's very important, I think, that this customization is allowed. That's really, really, really interesting. So one question I
Absolutely.
had for you is, what does the raw data from the analyzer, what does it look like? And what kind of post experiment data analysis is required to deliver a meaningful report to clients?
Sure. So that all varies on what the client is looking for. We will usually provide the client with the raw data. We perform a peer review of the work before we put it into a data summary that we provide in an Excel format file. And then from there, if the client is also interested in statistics, then we provide an average and a standard deviation for usually
Okay.
for each of the groups. And then if there's additional customization to statistics that they're interested in, we can always again discuss that prior to the study starting, or even upon, some interesting changes or findings mid-study, we can always
Right.
make those adjustments per client request. So
Got it.
we're
Got
extremely flexible here, which is something that when you and I have spoken in the past that I mentioned, whereas when you're in a GLP environment,
it.
sometimes
Right.
you are kind of confined to the rigidity of an SOP or a
Right.
protocol, whereas here at Biomere, we have some flexibility for non-GLP studies where we
Right.
can make decisions and changes on the fly, of course, provided a client and study director perform amendments, et cetera. But we are much more flexible
Yeah.
and able to go with the flow on most of our studies, which is a really nice feeling.
No, definitely. And we'll go into that a little bit more because I have some questions for you on, client behavior and
Mhm.
evolution of a study. So definitely we'll come back to that point. So I have to ask, I mean, like when I was looking at the different, clinical panels, chemistry panels that are out there, and I'm sure all of us have, been to the doctor, we've had blood work done and, you know, lipids or whatever, metabolic panels, what have you, CBCs. So tell me a little bit more about the panels that you guys typically run. Are they similar to what we see, in the clinical setting with patients, or are they very different?
They're exactly the same. So the
Okay.
analyzers that we have, the two primary analyzers, our Sysmex XMV for hematology, and then again the Beckman Coulter DXC 700 AU. Those are actually the same analyzers that they use over at UMass Memorial Health Center. These are
Okay.
two of the premier analyzers that are top competitive in the market for analysis in the clinical setting for human beings. So,
Amazing.
some of our medical technologists, in fact, we chose them and interviewed and brought them on board because of their experience with these analyzers. And
Got it.
so that experience is hard to find in the field unless you are
Right.
looking for a med tech, and often those med techs are coming from the hospitals.
Right. So this is truly a translational concept, right? I mean,
100%.
I think drug discovery is littered with a lot of, sometimes you have a lot of challenges where preclinical studies don't quite translate to the clinic for any number
Right.
of reasons. But this is absolutely amazing. So let's dig a little bit deeper on that. So, what requirements for clinical sample analysis? And I'm sure you've got a lot of experience in that space, of course. But it seems like when you're in a clinical lab, it's all about sensitivity, speed, and reproducibility. Go, go,
Mhm.
go. You're getting tons of samples.
Yep.
You've got to process them properly. You've got to get the data because, a lot of clinical decisions and healthcare decisions are made, based on these reports.
Yep.
So, is but preclinical is a bit different because you may be, kind of doing something a little different. So, in your mind, what key requirements for
Mhm.
to build, when you're in a preclinical setting where you're basically your samples are, rodents or NHPs or what have you,
Mhm.
um, are the requirements very similar for preclinical in terms of speed, reproducibility, and sensitivity, or what are the criteria do you look at? For example, is sample volume something
Mhm.
important?
Yes, so sample volume is important. It is going to be different based on the panel that the client is interested in. I think the biggest challenge with preclinical requirements comes with our small animal models, so the
Okay.
rats and the mice. And in those cases, we have ambitions, right? So we may have a client come in and say they want everything. So
Right.
sometimes where we have to go back to the drawing board and say, okay, this is or isn't feasible because of the size of this animal. And of course, Aya Cook is always the go-to for that definitive answer
Right.
as to whether this is ethical,
Right.
treatment of the animal. And so that's where, and you asked before about study models, that's where the study models can get really creative, either
Right.
adjusting the number of animals, when the necropsies and dosings are happening. Again, I have never been in a workplace that is so flexible and amenable to finding a study model that works for the client
Yeah.
when there are
Yeah.
limitations preclinically, and they want a certain
Right.
set of results. So
Right.
we almost always able to get all of that data for them,
Okay.
come to an agreement um, at some point pre-study where we can say, okay, we may not be able to give you all 20 of these studies from your teeny tiny mouse, um,
Right.
but we can give you a certain set of tests interim for interim time points that we can test for you. And then those will be very specific analytes that are going to tell you what's going on
Right,
with a particular organ system. And
right,
usually that will be relevant, of course, to whatever the TA is targeting or what they are anticipating for adverse effects from that TA. So
right, right.
it's really cool and exciting. I
Yeah,
came from a
I bet.
hospital environment and found that coming to research has always been my goal. I thought that I might be a PhD, and then I found the laboratory for clinical pathology, which I didn't even know was an option for
That's really
my
cool. Yeah,
certification. And so finding that was, I mean, I thought it was a gift from the stars for me,
I love it.
getting to come
Oh,
into
I love
that
it.
and then being on the research side, but still getting to do what I love. And
Right.
to be honest, I love that fast pace too. I love having that high throughput, getting a box of 100 samples and getting to see those results and putting it together in that data summary and delivering
Yeah,
it to the
yeah,
client. And then when
yeah.
we get to see the outcome of that in the news on occasion,
Oh my
I'm
goodness,
getting
yes.
chills just thinking about it. It's
Oh,
actually thrilling. I love it.
I couldn't agree with you more. I'm getting chills, and that's the beauty of it, right? Because what you just described is the vision, the big picture, right? Because I think what happens with a lot of when you're, in doing drug development, you get so fixated that on your it's very important to finish a study, get the data, but there's always, I think, a great idea to take a step back and be like, well, how is this fitting in the grand scheme of things? And
Right.
and it's very, very clear that this kind of analysis plays such a critical role because I mean, I always have been of the belief that well-designed preclinical studies not only accelerate drugs to patients, but it's the right thing to do. And
Mhm.
from an economic point of view, you actually end up saving time, you end up saving money because you were smart about your preclinical studies. And I just want to pause for a minute and acknowledge what you just said, which is around the thinking, the thoughtfulness that goes into the study, right? When, as you said, clients come and saying, I want all this data. And
Mhm.
what I'm really impressed to hear is the thoughtfulness that goes in. Because I'll be really honest, it's the easier option would be to say, nope, we can't do it. Sorry. Right? That's such an easy answer. But I think what is really amazing to hear is the fact that you guys are going above and beyond saying, okay, how are we going to make this work,
Yeah.
and within the biological limitations, because you can only take so much blood from a tail vein draw in a mouse for an interim time point. So I really want to just pause and acknowledge that thinking,
Mhm.
because that's truly being a collaborative partner and not just, hey, here's 100 samples, toss a
Yep.
box over the fence and say, get me the data.
Exactly.
I really, that's wonderful to hear. I
And
think that
you said
yeah.
collaborative, and that's exactly the word that was in
Yeah.
my mind. We make
Yes.
an effort on every study to collaborate from the client to the study director to the lab. And then we live and learn, right? So we may
Yes.
have a big ambition on a study. We try it. If it doesn't work, we know for next time, this is how we could
Yes.
do this better. So we are constantly improving based on that
Right.
experience. And again, in comparison with other environments that I've worked in, we didn't have that flexibility. You either did it
Right.
or you didn't do it. And they weren't
Right.
willing to make those efforts to try
Right.
something new, to try something that might be a little more effort or work or labor.
Yes.
Those things were just a flat no. So I
Right.
love that we really do make that effort to collaborate, to try things, to think out of the box, to get the results that the client wants. And I think that kind of teamwork and collaboration is necessary for the
Right.
kinds of innovation that, you're looking for in the pharmaceutical industry.
Completely
Everything is
agree.
competitive and everything is about, getting that result first. And sometimes
Yes.
you have to do things a little differently to do that. So at Biomere, we definitely, that's literally
Yeah.
our philosophy.
I love it.
We will
I
give it
love
a
it.
try.
No, that's a beauty, and I think that kind of is a is a full circle back to the question of, you know, some of the key differences between clinical and preclinical studies, and you articulated it beautifully in the sense that you do have that flexibility, whereas in clinical, it's kind of this is it. You
Yep.
can't change it. You
Mhm.
and many people can't, won't, don't want to, whatever.
Yep.
Whereas in preclinical, you've got that flexibility, and essentially at the end of the day, what you're looking for is actionable data to inform
Right.
the next step. So that's actually a perfect segue into, you know, revisiting something you'd mentioned earlier, which is around, you know, the changing study designs based on the data. Sorry. Yeah, so you know, going back to the changing of the study designs based on the data. So since this is, you know, non
Mhm.
studies that a lot of what you're doing, how does, you know, how often do you see clients, you know, changing the study? And then I'm going to ask a little cheeky question. In your experience, how does an original, you know, study that a client signed off on, your original proposal, and the end study, how much variability do you see in that on average?
Um, I would say that it is, I'd say 50-50. So
Okay.
it all depends on the test article. And it all depends on where they are at in the development of that test article. So, say you have a test article that is very new. It's late discovery. You're
Mm,
still working on refining that test article. Um, those studies can be a little messy where
okay.
you find that maybe the animals aren't responding as well as you
Okay.
like, um, or maybe it's not a health concern, but just you're not seeing the results that you were anticipating. So when you get those early or late discovery test articles, there can be a lot of variability. And then other clients we have some very refined, specific test articles. They're looking for something and they have it already keyed
Ah,
in. Those studies are just, we get the study protocol, we run the time points, we deliver the
right.
data, easy peasy. So
Right.
we really do have a good, I'd say probably 50-50 mix of both. Um, Oftentimes, the other challenges that we have, so again, you have the large animal model and the small animal model.
Mm-hmm, mm-hmm.
Um, you may have a test article for a small animal. Well, the difference in a clinical outcome, a critical clinical outcome in a mouse and an NHP is huge.
I bet. Yeah.
Ethically, that mouse may have to be necropsied,
Right.
in the name of ethics, whereas in NHP, we can provide different kinds of support to that animal, so that they're comfortable and that we can proceed with the study and still get the results that we planned on.
Right. Right.
So we really, it's 50-50. It depends on the clients that are in the building at the time. And we just roll with it.
I love that. No, that makes so much sense. And what you know what I'm taking away from this is that it seems like depending, you know, where the client is in the drug development process, as you mentioned, it's good to hear that there's, again, we come back to that favorite word, flexibility.
Mhm.
Of, you know, it seems like your lab is flexing because if it's an early stage client, they're not really sure what's going on, don't really have as much data locked in, or still there's a lot more risk and uncertainty,
Yes.
right, in the
Yep.
study. You guys can flex to handle that, but you can also flex to say, you know what, no, no, nothing needs to be tweaked big time. We just need to run the
Right.
samples and with speed, get the data and move on. So that I totally
Mhm.
get that.
Yes.
I'm going to segue a little bit, Atia, into asking kind of like some of the logistics questions. So, the questions. So typically, when you're doing samples, you know, especially interim dosing, right? I'm sure you have an option of either running the samples right away, so fresh samples, or freezing them. So, in your experience, do you see a big difference in, analyte readout consistency and quality between fresh and frozen samples?
Not at all. So, particularly with the Beckman Coulter analyzer, and to be clear, hematology specimens are run within 24 hours of collection when
Okay,
feasible.
okay,
And then clinical chemistry specimens, so serum, plasma, etc., those are almost always able to be frozen with
okay,
very few exceptions. There's maybe one or two analytes where you will see a difference if it is not a fresh specimen, but I would say 99 out of 100
okay.
analytes we can freeze and analyze at a later date. The deeper the freeze, the more preservation of those analytes. So we always
Right.
freeze at -80.
Yep.
Even if the recommendation is -20, the deeper the freeze, the more stable. So
Right.
those samples, we've had clients, send us stuff that's a year old,
Yep.
where we still go ahead and are able to analyze. And
Okay.
there's also, so every analyte that we use on the Beckman Coulter analyzer has a stated stability that's been studied as well. And
Okay.
we do adhere to those guidelines when we are performing analysis of samples that have been stored for a certain duration of time. And if it exceeds that, we let the client know. And,
Okay.
take that with,
Yeah.
you have to appreciate that for what it is. There's no way to know
Yes.
what the effect is, what the impact is.
Right. Yeah.
But every analyte on our chemistry analyzer has a stated stability that's been studied and verified as well. And
Nice.
it's usually
Nice.
several months, years of
Yeah.
storage is allowed.
So Tia, I just want to pull on that thread a little bit more. So, because you talked about client samples, I was always under the impression that, most of the samples that were analyzed in your lab were from were taken from animals in, where studies were taken from animals, from at Biomere, so studies being run at Biomere. But it seems like you're also doing client samples. So if that's true, what's the distribution of, in-house samples versus client source samples?
Sure. So, to be honest, about 95%, I would say, of our samples are based or delivered from studies that are being performed with the in vivo in-house. But about 5%, and we're still growing. Sometimes we go up, sometimes we go down. About 5% of those samples are shipped to us from clients, where, we've had a few very interesting examples where we had a client that used to do all their in vivo in-house full study, start to finish. And then they had some budget changes. So they began to do their own in vivo, and then they were shipping samples to us. And then that same client, in fact, had a situation where something came up, a new question about a test article. And so they had some frozen specimens that they were able to ship us. And in fact, we actually had a new analyte that we had just added to our menu that suited what they were interested in looking at. So it's pretty exciting that we were able to, pivot based on what the client needs in the moment, because, budgets change,
Yeah.
directions change, et cetera.
No, and I mean, I think that, I'm glad you're bringing this to the conversation because it's such an underrated service, right? Because again, this is where the collaboration, the flexibility comes in, right? To account for budgets, to account for, what the client needs are. And it's really interesting that, a client could do the in vivo studies, the dosing, the sampling, but what an advantage for them to be able to ship samples quickly to a CRO, which has top of the line analyzers and who've got the workflow locked in, all the way from sample
Absolutely.
receipt, processing, analysis, data. And what a luxury it is for clients like that to be able to do that without, and have that flexibility. And I think that's something that's so underrated.
Mhm.
And I think I'm glad we're bringing it to the surface here because this again gives clients flexibility. And again, budgets come, budgets go. Sometimes you can outsource the whole study, sometimes you can only outsource some of the study. So
Right.
this modular approach, I think, is really, really wonderful. So
Definitely. And sometimes too, you only have one shot, right? You have a small
exactly,
amount of
exactly.
sample. So for example, that last scenario that I explained, they sent us some samples for analysis. And, maybe it is something they could work on themselves or sort it out. But if you only have 20 microliters of sample left to work with, then you want it done right the first time. You don't want to roll the
Completely.
dice.
Yeah. And in science, sometimes you get do-overs, sometimes you don't. And in vivo, when especially with the mouse study, this is it. That's all you can
Yep,
get from a tail vein, and you kind of have to make it work. So
exactly.
completely agree with you. I'm so glad we brought up this point. So I'm gonna ask you one last, question. And I know that a lot of the clinical chemistry, you have like a lot of standard panels, like we talked about. But it also seems that people want to add more and more unique markers for their studies and things like that. So, do you have an example where a client, kind of introduced a new marker or just kind of went in a different direction from your standard, chemistry panel?
Definitely. So actually within the past few months, we had a really interesting situation. We were planning to bring a particular analyte on board on our clinical chemistry analyzer, and this client happened to also be interested in that. So when we had our first meetings with the client, they described what they were interested in doing. They wanted to um, pretty much repeat some research that was published in Nature magazine. And they had a similar test article, same target. And so when we had that conversation, they decided that they wanted it to be as close to that original study as possible. But then they also wanted to improve on that study model as well. And so what we ended up doing was we tested out two or three different reagents or vendors for that analyte. And then we provided that data to the client with the test run. And so they were able to take that data, decide which vendor they wanted to use for the study, and go from there. Once we did that, they also decided that they wanted the results reported in two different units. And this was particularly challenging for this analyte because the target is a lipid. And so the method of testing, I believe, is immunoturbidimetric. So that's based on
Okay.
an actual solid particle in your sample. And so it
Okay.
wasn't just as simple as converting units using a conversion factor. It is actually measured in a slightly different way
Oh,
based
wow.
on the units of reporting. So what we had to do, and this vendor supported this in their own research and validation of that reagent for that analyte, we validated both sets of units for the client, and then we ran the tests and reported the tests in both sets of units for the client. So
Wow.
something like that, being able to do the work ourselves, validate the test ourselves, and then provide the very specific requests that the client was interested in was really exciting for me. It was my first time doing anything like that. But we definitely made it work. And now on the flip side of that, we're able to offer it to any other client that is interested in
Right.
performing that kind of testing as well.
And I think it also gives you the blueprint, right? On
Yes,
how to do
exactly.
this. And so the next time you'll get a request, you guys, how to kind of do the validation, what
Yep.
the steps are. And
Yes.
as you were telling me the story, I was thinking, wow, this is just going above and beyond, right?
For sure.
And so I think, it's a good idea to pause and just think, when people think clinical chemistry, right? They think standard panels, blood draw, whatever, you get the results, get a report, you move on. But I think it's really a good time to really be very clear and say, wow, this is development, this is R&D,
Yes,
right, of
definitely.
methodologies of adding new analytes. And I just want to emphasize the fact that Biomere may be a CRO, but it's really really good to hear about the innovation, the
Yes.
R&D, supporting client needs. And I think and just advancing the capabilities a little bit more, a little bit more.
Mhm.
And I think with every study. So That's wonderful. That's absolutely
Thank
wonderful
you.
to hear. And on that note, I will say thank you so much. This was a
Welcome.
phenomenal discussion. I think there's so much good information that came to the surface, and I know I learned a lot. So I'll say this to our listeners: if you would like to get more information on Biomere's portfolio, please email info@biomere.com, and please feel free to check out the website biomere.com, as well as you can check out in the website there is a section called the concourse, where you can, it's a portal that houses a ton of technical resources, podcasts, blogs, technical papers, video recordings, what have you. So it's a great place to learn different techniques and methods. So with that, Tia, I will say thank you so much for joining me today, and I
You're
really
welcome.
appreciate all the knowledge that you've shared with us.
Thank you. Thank you for having me. Take care.